Alright, we are going to start talking about neuroplasticity, a very interesting area of the nervous system. So basically, neuroplasticity is the ability of neurons to change their function, their chemical profile, or their structure. And the chemical profile is the amount or types of neurotransmitters that are being produced, or any other chemicals that they're being produced. So the state of our body, of health or disease, or what new experiences we are going through, continuously create and break neuronal communication sites. So the neurons themselves have to change to accommodate what is going on in our body. And the process by which they do that is called neuroplasticity. So neuroplasticity is involved in learning and creation of new memories. It's essential for recovery from damage to the central nervous system. So by definition, neuroplasticity is change that has to last longer than a few seconds, and it's not periodic, so it doesn't come and go. So the general term neuroplasticity is used to encompass the following mechanisms: habituation, and we'll talk about each of these individually; experience-dependent plasticity, or learning and memory; and cellular recovery after injury. So habituation is the simplest form of neuroplasticity, and it's a decrease in the response to a repeated benign stimulus. So in the 1800s, there was a neurologist named Charles Sherrington. He did a set of studies where he observed that certain reflexive behaviors, like withdrawing a limb from a mildly painful stimulus, ceased after several repetitions of the same stimulus. So his animal subjects were getting used to that mildly painful stimulus. So that was sort of the early study of habituation. We use techniques and exercises in PT and OT intended to decrease the neural response to a stimulus. So if you have a patient that is extremely reactive to stimulation on their skin, a lot of times after surgery, the surgical site is extremely reactive. Sometimes in children it's called tactile defensiveness. But by gently stimulating the skin and then gradually increasing the intensity of the stimulation, it's intended to achieve habituation to the tactile stimulation. So also people with certain types of vestibular disorders that induce dizziness and nausea, we are repeating the movements that induce those symptoms repeatedly, and the purpose is to achieve habituation to the movements. So we use habituation a lot in therapy, particularly if you are, particularly if you're working with human beings, because all of the things we do are about learning and getting used to things or changing things. So experience-dependent plasticity, it's also known as activity-driven neuroplasticity. It's a complex process involving persistent, long-lasting changes in the strengths of synapses between neurons and neural networks. So when you're learning a new task, you have to build the new neural network to accommodate that new task. That is what the learning process is. So a good example is learning to play a musical instrument, or learning a new language requires numerous brain regions, not just one. So during the initial phases of any motor learning, large and diffuse regions of the brain are active. When the tasks are repeated, the number of active regions are reduced. And when a motor task is mastered or fully learned, only a small distinct region of the brain shows an increased activity when performing the task. And it's sort of like when you're learning how to drive, you really have to, keep your hands at 10 and 2 and look straight ahead, and you can't be distracted. But then once you've really learned it, you've mastered the task, you can drink coffee and eat a sandwich while you're driving, hopefully safely when you do that. Okay, I admit it, I spill coffee in the car a lot. This is why I don't drive most of the time. But, so when a motor task is learned, you need to use less of your brain to do it. So that's kind of cool, actually. So the mechanisms that are involved in experience-dependent plasticity depend on the type of synapse and the location involved. So the mechanisms include plasticity of the intrinsic excitability of the neurons by functional changes in ion channels. So we talked a little bit in the last section about synaptic receptors that cause a change in gene expression. So those ion channels are proteins that are coded for in your DNA. And so if you're getting different input, you could potentially be changing the kind of ion channels that are required. And that's part of how the neurons change their chemical profile. So there's plasticity that's inhibitory at GABAergic synapses, so synapses that are producing GABA. There's homeostatic plasticity to stabilize neural circuits. So basically when we build a new neural circuit, we have to make sure we have the chemical and physical infrastructure basically to maintain and stabilize those neural circuits. So long-term potentiation and long-term depression of different synapse sites, and we will discuss those further next. So long-term depression is long-term potentiation and long-term depression are changes basically in neurotransmitter release or changes in receptor density and efficiency. So when we look at, these are the figures from the book, and when, so the first one is long-term potentiation, the other one is long-term depression. So basically we have some receptors that are available in what's called a silent synapse. And a silent synapse, it's not being used, basically. And so when you start to stimulate that synapse, the receptors that are being built in the cell basically migrate to the synapse, and they provide more receptor sites. So you actually get transformation of the synapse with repetition of a task, which is kind of neat. And it's just the opposite in long-term depression. If a synapse isn't used, it can become silent. You're using fewer of those receptors, and so the cell starts to break them down and not use them. So different forms of long-term potentiation and long-term depression can occur simultaneously depending on the type and the location of the synapse. So the interesting thing is that plasticity can be maladaptive. It can have harmful consequences. It may contribute to the development of chronic pain syndromes, including low back pain. So if you're, say that you're super sensitive to a particular stimulus, your neurons might say, oh, I need more receptors for that stimulus, even if it's one that's not doing you any good. So long-term potentiation and long-term depression can definitely kind of shoot you in the foot on some of those things. So you can get used to anything, and you can get used to having increased levels of pain. So in the book they talk about transcranial magnetic stimulation, which I've never seen or experienced, but it's interesting. Depending on the frequency and experimental protocol used, it inhibits motor learning, and either enhances or inhibits motor learning and memory formation. So they use it to induce a transient virtual lesion to assess the impact of different brain areas have on motor learning. So that's pretty neat, because in the old days, before they had this, they used to have to wait until somebody just had that lesion to figure out what the impact was on motor learning, and then you couldn't get it back as easily. So the magnetic stimulation of the brain is thought to induce synaptic plasticity either via long-term potentiation or long-term depression type mechanisms. So that's kind of interesting. I imagine that's done in an experimental setting, but there's a picture of it in the book too that looks pretty cool. So astrocytes contribute to experience-dependent plasticity. So communication between astrocytes and neurons occurs via the release of a neurotransmitter by the neuron that stimulates the release of gliotransmitters by the astrocyte. So gliotransmitters modulate neuronal activity and synaptic transmission. So astrocytes likely influence synaptic plasticity through modulating neurotransmitter release and receptor expression at the postsynaptic membrane. So that's kind of neat. So astrocytes may also be important for new synapse formation following a stroke. So they actually play a critical role in brain and spinal cord plasticity. So the more we find out about the different cells, the more we find out about potential for recovery. So long-term changes, including synthesis of new proteins and growth of new synapses, result in a maintained response and memory of specific repetitive stimuli. So that's pretty neat that the astrocytes have an effect on that. So I'm going to wrap this part up, and then we're going to start talking about metabolic effects of brain injury and recovery from central nervous system assaults.